Welcome to the Introduction to Food Macromolecules simulation. In this simulation, you will learn about the different types of macromolecules found in food, carbohydrates, protein and fats. This simulation uses voiceovers. If you don't have audio, click the speaker symbol on the top right of your lab pad to switch the play mode. If you disable the sound effects, the lab pad will slide up whenever there are instructions to read. You can select what type of audio you prefer. Deselecting any of the options will mute that option. Click Continue to learn how to navigate through your mission. Using the lab pad is an important way to get the most out of this simulation. Is this the first time you've played a Labster simulation? Bob and Alice are partners in a biology class. After they've been working together for several weeks, Bob decides to ask Alice for a date in the college cafe. Exciting.
Wow, you sure seem to love cabbage salad.
Yeah, it's delicious.
I wonder if we could survive only on cabbage salad. Does it have all the important macromolecules we need?
Cabbage salad definitely has great nutrients, but I'm not sure if it has all the macromolecules we need.
Wanna take this date to the lab and maybe we can find an answer together.
I do love nerds, let's go.
Hi, I'm Tracy, a food scientist. I'll be your assistant today. Molecules in food can be divided into two main groups, micro and macro. Micromolecules include vitamins and minerals. Macromolecules include carbohydrates, proteins, and lipids. In this lab, you will learn about and perform a test for each of the macromolecules. Macromolecules are large molecules. Macro means large, and molecules refers to atoms held together by chemical bonds. Which of the following molecules is not a macromolecule? You are correct. In biochemistry, we know four types of macromolecules: carbohydrates, proteins, lipids, and nucleic acids. In food, we are mainly concerned about carbohydrates, proteins, and lipids. Let's start by taking a look at a few different types of food.
Take a look at parts of Alice's and Bob's meals at Workbench 2. Before we get started, you'll need to put some gloves on by clicking on the glove box. There are three macromolecules found in food: carbohydrates, protein, and fats. You can see them all in front of you here. Look at all this food. Are we allowed to eat it after the experiment? Yes. In the lab, you may be exposed to hazardous biological and chemical materials through consumption of potentially contaminated food and drinks. To avoid any risks, you shouldn't prepare, store, or consume food or drinks in the lab. Click on the food that contains a high amount of carbohydrates. Yes, carbohydrates include simple sugars and complex sugars. They are the source of energy for our body. They can be found in both healthy and unhealthy food, such as rice, bread, popcorn, cookies, pasta, apple pie, milk, potatoes, and soft drinks. Click on the food that contains a high level of protein. Bingo! Protein is found throughout our body. Protein builds our enzymes, cells, hormones, and antibodies. If your body lacks protein, you will start experiencing loss of muscle mass, weakening of the heart and respiratory system, and decreased immunity. High-protein food includes fish, chicken, beans, nuts, and red meat. Click on the food that contains a high amount of lipids. You know your foods. Lipids include fats. The major function of fat is to store energy. It also maintains your core body temperature. Fats are mainly found in oil, butter, cheese, and processed foods such as pastries, cakes, and biscuits. Delicious time to make a hypothesis. We are curious about Alice's cabbage salad. What type of macromolecules do you hypothesize to primarily find in Alice's salad? Alice's salad mainly consists of cabbage. You can perform a series of biochemistry experiments to determine what type of macromolecules are most common in this salad. Some macromolecules are polymers. What is a polymer? Awesome! A polymer is a long molecule that consists of small repeating units called monomers. You can imagine a polymer as a train with the individual monomers as the cars. We will perform 4 biochemistry tests to elucidate the macromolecule content of Alice's cabbage salad. We will also check other food samples for their carbohydrate, protein, and fat content. See the following workflow. You can see the workflow image anytime from the Media tab. Go to Workbench 1, Carbohydrates. Let's begin with our first experiment. At this workbench, we'll test Alice's salad for carbohydrates. Carbohydrates provide the body with energy. They're often known as sugars or saccharides. Carbohydrates are commonly divided into simple sugars and complex sugars. Click on the syrup to jump into an animation about simple sugars and their chemical structure. Carbohydrates are organic compounds that are composed of carbon, oxygen, and hydrogen. Their empirical formula is CH2O, which is virtually a carbon and water, or hydrated carbon, hence the name carbohydrate. Carbohydrates include sugars and polymers of sugars. The simplest type of carbohydrates are monosaccharides or simple sugars. Monosaccharides or simple sugars can be aldose or aldehyde sugars, as shown on the left, or ketose or ketone sugars, as shown on the right. It depends on the location of the carbonyl group. Which of these is an aldehyde? You're a chem whiz. An aldehyde has one alkyl or aryl group and one hydrogen bonded to the carbonyl carbon. A ketone has two alkyl or aryl bonded to the carbonyl carbon. This functional group determines the type of monosaccharides. Monosaccharides can exist as a linear chain or as ring-shaped molecules. This is the linear chain shape of glucose, an example of a monosaccharide. In aqueous solutions, monosaccharides are usually found in ring forms. Monosaccharides can link together, forming oligosaccharides. Oligosaccharides with 2 sugar monomers are known as disaccharides, with 3 as trisaccharides, with 4 as tetrasaccharides, and so on. This is sucrose, an example of a disaccharide, which is a combination of 2 monosaccharides, glucose and fructose. What forms after the glycosidic linkage reaction between glucose and galactose is complete? Splendid. Glucose and galactose combine via glycosidic linkage, forming lactose and water. Lactose is a sugar mostly found in milk. Glucose and fructose form a covalent bond between them. This bond is called a glycosidic linkage. Both glucose and fructose are hexoses, though fructose forms a 5-carbon ring, while glucose forms a 6-carbon ring. Remember that monosaccharides are the same as simple sugars or simple carbohydrates. We will discuss complex carbohydrates later. Great! You covered the basics of carbohydrates in the animation. To sum up, there are 2 types of carbohydrates, simple sugars and complex sugars. Benedict's reagent is a chemical reagent that detects the presence of simple sugars, monosaccharides and most disaccharides. Table sugar cannot be detected using this reagent. Let's see if Alice's salad contains simple sugars. Add 1 milliliter of Benedict's reagent to each tube. In a real lab, you have to repeat this for every tube. Luckily, Tracy will help you out with this. Now put the dropper back into the bottle. Pick up the first tube containing Benedict's reagent using the test tube holder. Place the test tube in the boiling water on the hot plate and then wait for 5 minutes. Click on the lab pad for more information. Alright, 5 minutes is up. Take the tubes out of the boiling water on the hot plate. Place the test tube holder back on the table. The presence of simple sugars is indicated by the change of color. In this Benedict's experiment, the positive control is glucose, and the negative control is water. Slide down the lab pad or click the View Image button to examine the color of the food samples. Click on the View Theory button for more information. Benedict's reagent detects the presence of simple sugars in food. What color indicates the presence of a high level of simple sugars? Spot on. Benedict's reagent is used to test for glucose but not sucrose. In the experiment, the test sample is heated and analyzed for color change. A change from blue to red or orange indicates a high level of simple sugars in the food sample. A low level of sugar is indicated by the colors yellow and green. Blue means no simple sugar is present in the food sample. Click on the tube that contains only a low level of glucose. Raise the lab pad or click on the View Image button for help. Yes, you have a sweet tooth. Examples of foods that contain a high level of simple sugar are cookies, cakes, soft drinks, honey, milk, and fruits. Simple sugars are digested quickly by the body and enter the bloodstream quickly. Having excess sugar in your blood can contribute to gaining weight and developing type 2 diabetes and heart disease. Great job completing Benedict's test. Next, you'll complete the test for complex sugars. Click on the baked potato to jump into an animation about complex carbohydrates. Potatoes are an example of food with a high carbohydrate content. Remember that carbohydrates includes monosaccharides and complex carbohydrates. In this animation, you will learn the basic structure of complex carbohydrates. This is the structure of starch, an example of a complex carbohydrate mostly found in plants. Complex carbohydrates are polymers called polysaccharides that are composed of many sugar building blocks. Which of the following is a carbohydrate? You got it. Sucrose is part of the carbohydrate group. DNA is a nucleic acid. Insulin is a peptide hormone, so it's a protein. Wax is part of the lipid group. The sugar building blocks are monosaccharides, such as glucose. The glucose monomers in starch can also be joined by 1 to 6 linkages, forming a branch in the structure. There are two types of starch, amylopectin and amylose. Amylopectin is branched, while amylose is unbranched. As you might know, plants store energy as starch. You can see amylose at the top and amylopectin at the bottom left in front of you. Animals store energy in a similar structure called glycogen, which you can see in the bottom right. Now take a look at broccoli. It has a crunchy structure thanks to another complex carbohydrate called cellulose. Unlike starch, whose main function is to store energy, the main purpose of cellulose is to maintain cell structure. Even though starch and cellulose have different functions, their structure is pretty similar. Cellulose is also a polymer of glucose. The difference between cellulose and starch is in the type of glucose building blocks. In starch, the glucose is an alpha glucose, while in cellulose, it is beta glucose. That's why cellulose is mainly straight, while starch is helical. The figure below shows the structure of alpha and beta glucose. What is the difference between the two? Genius. The alpha and beta glucose differ in the position from the hydroxyl group attached to the number 1 carbon. The hydroxyl group is upside down when we compare between the two glucose structures. The figure below shows the structure of alpha glucose and beta glucose. So the glucose in starch is an alpha glucose while the glucose in cellulose is a beta glucose. Humans have the enzyme to break down the glycosidic linkage in starch but not in cellulose. That's why we can't digest the cellulose in vegetables. Nutrition facts labels tell you the amount of macromolecules in the food. What does insoluble fiber refer to? You've got that. Insoluble fiber refers to the type of carbohydrate that your body couldn't digest. A human doesn't possess the proper enzyme to break down cellulose. Food that contains high level of cellulose are brown rice, whole wheat bread, fruits, and vegetables. Awesome! You covered the basics of complex carbohydrates. So, complex carbohydrates have longer chains of sugars than the simple sugars. The chains can have branches or not, depending on their origin and function. Iodine solution is a chemical reagent that detects the presence of starch. The positive control of this experiment is a potato and the negative control is water. Add drops of iodine reagent to each tube on the right-hand set of test tube racks. In a real lab, you have to repeat this for every tube. Luckily, Tracy will help you out with this. It's that beautiful science time. Put the dropper back to watch the colors change over time. Click on the tube that contains food with a high amount of starch. Click the Lab Pad or View Image button for help. Marvelous! Iodine reagent interacts with and binds to a structure in the starch molecule, forming a structure that has a dark bluish-black color. Therefore, food that contains a high level of starch, such as potato, will exhibit a black color, whereas food that doesn't contain complex carbohydrates will exhibit a yellowish color. What do you call a sample, for which no response is expected? You are correct. The negative control is essential to check that the positive result is actually positive, and not because of an error in the procedure. You can observe the results of the Benedict's test for simple sugars and the iodine test for complex sugars using the Media tab. Now it's time to input and assess the results in the test results table. See the following workflow. You can also see this workflow image anytime in the Media tab. Fill out the test results table correctly on the screen. Great job! Now let's continue to the next mission. We will make a final assessment of Alice's cabbage after we performed all 4 biochemical tests. Proteins are organic molecules made up of amino acids. It's important to consume enough protein. Our body needs it to produce important molecules such as enzymes, hormones, antibodies, neurotransmitters, and parts of the cell structure. Good sources of protein are eggs, beans, milk, fish, and meat. Click on the eggs to dive into an animation about proteins. Watch the animation. Genetic information is stored as DNA and copied as messenger RNA during transcription. In mRNA, genetic information is encoded in 3-letter units, codons, made up of the bases uracil, cytosine, adenine, and guanine. In the ribosome, each codon is translated into 1 amino acid. Amino acids are linked by covalent peptide bonds. The formation of a peptide bond produces 1 water molecule. Amino acids are linked by a specific type of covalent bond. The reaction producing this bond also produces 1 water molecule. What type of bond links amino acids together in a chain? Smart. A covalent peptide bond links 2 amino acids, thereby forming a long chain. Peptide bonds occur between the amino group and the carboxyl group of neighboring amino acids. The process of making a peptide bond results in 1 water molecule. An amino acid molecule consists of 3 main components, a carboxyl group, an amino group, and a side chain symbolized by R. A peptide bond occurs between the amino and carboxyl groups of neighboring amino acids. A sequence of amino acids, called a polypeptide, grows from the N-terminus to the C-terminus. This polypeptide chain represents the primary structure of a protein. A sequence of amino acids, or a polypeptide, grows from the N-terminus to the C-terminus. The linear sequence of amino acids within a protein is considered the structure of the protein. Nice. The primary structure of a protein is the linear sequence of amino acids. Which level of protein structure is formed by the weak bonds between oxygen and hydrogen atoms within the polypeptide backbone? Smart. The oxygen and hydrogen atoms within the polypeptide backbone can form hydrogen bonds. These weak bonds result in the local folded structure known as the secondary structure. Which of the following is a secondary protein structure? Perfect. Two basic types of secondary protein structures are alpha helixes and beta sheets. The alpha helix is a right-handed coil. The beta sheet can be formed by two or more segments of the polypeptide chain lying side by side. The entire 3D structure of the folded protein is known as the tertiary structure. Proteins are modified in many ways after translation. Here, you can observe a protein that undergoes post-translational modification, called glycosylation, before it can be considered biologically active. Glycosylation is the addition of sugars. Now you know that proteins consist of long chains of amino acids. The chains are folded up in specific ways to become functional. The burette reagent will reveal the presence of protein. A positive result is indicated by changes in color from blue to dark purple. Add 0.5 ml of the burette reagent to each tube. In a real lab, you have to repeat this for every tube. Luckily, Tracy will help you out with this. Put the dropper back into the bottle, and then watch the colors change. A positive control is a sample known to give positive results for the given test. What is the purpose of a positive control? Great work. Controls are needed to make sure the procedure is working correctly. A positive result from the positive control, even if the samples are negative, will indicate the procedure is optimized and working. It will verify that any negative results are valid. If there is no positive control and the sample is negative, we can't be certain whether the sample was truly negative or if the procedure didn't work. Click on the positive control of the burette assay. Raise the lab pad or click on the View Image button for help. Click on the lab pad for more information. Great job! The positive control for the burette experiment is an egg. Eggs are high in protein content, therefore they will exhibit a purple color after this biochemistry test. Water, on the other hand, the negative control, will exhibit a blue color. Now that you've completed the burette test for protein, let's assess the results, just the way you did with the carbohydrate biochemical test results. See the following workflow. You can see the workflow image anytime from the Media tab. Fill out the table of biochemistry test results correctly for the burette test. Great job! We can use these results to help make a final assessment of the macromolecules in Alice's cabbage salad. Go to Workbench 3 to begin investigating lipids. You've tested the food samples for carbohydrates and protein. Now let's check whether it contains any lipids. Lipids are a group of molecules that do not mix well with water. Lipids include fats, sterols, and phospholipids. When it comes to food, we focus on fats. Fats provide insulation, energy and a cushion for organs. Good sources of fats are avocados, fatty fish, egg yolks, and vegetable oils. Click on the butter to dive into an animation about fats. Lipids are not true polymers. Lipids include fats, phospholipids, steroids, wax, and certain pigments. These compounds are grouped into a class of molecules called lipids because they share one important trait: they mix poorly with water. Fats are composed of two kinds of smaller molecules, glycerol and fatty acids. Glycerol is an alcohol. Glycerol has 3 carbons, each of them bears a hydroxyl carbon. Fatty acids have a long carbon skeleton. The acid in fatty acids refers to the carboxyl group at one end of the carbon skeleton. The rest of the skeleton consists of a hydrocarbon chain. This hydrocarbon chain is hydrophobic, which means it separates from the water. Each fatty acid is linked to the glycerol by an ester linkage, resulting in a fat, also called a triacylglycerol. Which type of molecule are lipids mostly made of? Impressive. Lipids are organic molecules that are insoluble in solutions of high polarity such as water. For example, think about interaction between oil, which is a lipid, and water. Lipids are insoluble in water because they are largely composed of nonpolar regions. Nonpolar regions are hydrophobic. The figure above shows the structure of fatty acid. Is a large nonpolar region and is a small polar region. Saturated and unsaturated fats refer to the structure of the hydrocarbon chains of fatty acids. Saturated fats have no double bonds between the carbon atoms. Hence, the structure is saturated, with hydrogen. Unsaturated fats have one or more double bonds between carbon atoms. The hydrocarbon chain will bend at the position where a double bond occurs. At room temperature, the molecules of saturated fat are packed closely together, forming a solid. The molecules of unsaturated fats cannot pack together tightly enough because of the kinks in the fatty acid hydrocarbon chains. A trans fat is unsaturated fat that is uncommon in nature. It is formed when unsaturated fats are synthetically converted to saturated fats by adding hydrogen. Which of these is the structure of trans fat? Remarkable. Trans fatty acid or trans fat is an artificial unsaturated fat. It contains a trans double bond between the carbon atom. The figure below shows the structure of saturated fatty acid, unsaturated fatty acid, and trans fatty acid. So you can find unsaturated fat in oil, saturated fat in butter, and trans fat in margarine. Super nice. Now you know the basics of lipid structures. Basically, fats are made up of glycerol and fatty acids. The fatty acid chains can be saturated, unsaturated, and trans-unsaturated. Sudan 4 is a red stain that detects the presence of lipids. When Sudan 4 is added to a mixture of lipid and water, the dye will move into the lipid layer, coloring it reddish-orange. Fill a tube up halfway with water using the pipette. In a real lab, you have to repeat this for every tube. Luckily, Tracy will help you out with this. Put the pipette back in the holder. Mix the contents of a tube using the vortex. Put the tube back into the rack when the vortex is done. Tracy has vortexed the rest of the tubes for you. Now you should add drops of Sudan 4 to each tube. In a real lab, you have to repeat this for every tube. Luckily, Tracy will help you out with this. Put the dropper back into the bottle. Mix the contents of a tube again using the vortex. Place the tube back on the rack when the vortex is done. Click on a test tube containing a food sample with a high fat content. Raise the lab pad or click View Image for help. You are correct. Foods that contain high level of fats are butter, oil, milk, and eggs. Splendid work so far. You've tested the presence of carbohydrates, protein, and fats in various food samples. Ultimately, you're close to making a conclusion whether Alice's salad contains all of the macromolecules or not. Let's input our result in the laptop first. Fill out the table of test results correctly for the Sudan 4 test for lipids. Congratulations! You have completed tests to identify all food macromolecules correctly. Well done, foodie. Now we're ready to make a conclusion. Just before that, let's quickly test your ability in recognizing macromolecule structures. Go to the molecular viewer to view the structure of a few macromolecules. Which macromolecule is shown here? Click on the correct name on the molecular viewer. Well done. 2 more to go. Great. Which macromolecule is this? Click on the correct name on the molecular viewer. Well done. The last one is coming up. Yes. Which macromolecule is this? Click on the correct name on the molecular viewer. Congratulations. You know the structures of these important food macromolecules. Learn more about macromolecules by clicking View Theory. You are ready to go to workbench 2 to make a conclusion about Alice's cabbage salad. Alright, time to put together everything you have learned. Slide down the lab pad to see the table on the screen or click on the View Image button. Which food contains carbohydrates, proteins, and fats? Nice. Based on the biochemical tests, only milk contains a complete set of macromolecules, with the exception of complex sugars. Drinking milk every day will supply you with all the macromolecules and minerals your body needs to function properly. Which sample contains no macromolecules at all? Correct. Water does not contain any macromolecules. That's why we used it as the negative control in our 4 biochemical experiments to detect macromolecules. Despite the lack of macromolecules, it is essential that you drink enough water to keep your body hydrated. Earlier you made this hypothesis: Alice's cabbage salad mostly contains simple sugar. Lower the lab pad to examine the result of the biochemical experiment that we documented on the laptop or to take another look at the tubes on the workbench. Is your hypothesis true? Genius. We've conducted all 4 biochemical tests, 1 each to detect simple sugar, complex sugar, protein, and carbohydrates. Of the 4 tests, 2 came out with positive results, detecting simple sugars and complex carbohydrates. The level of simple sugars detected was, however, very low. Based on the iodine test, Alice's cabbage salad contains a high level of complex carbohydrates, but it lacks the other important macromolecules, proteins and fats. Based on the biochemistry tests, Alice's cabbage salad lacks some important macromolecules. What should Alice add to her salad to provide the missing macromolecules she needs? Good call. Alice's salad lacks protein and fats. She can add an egg as a source of protein and an avocado as a source of fats. Let me ask you one last question. Do you feel like you gained relevant knowledge during this simulation? Thank you for your feedback. Your score has been saved together with your 100% progress. Congratulations, you have completed the simulation. We hope you enjoyed it and you feel wiser now.